We were discussing about epitope. Okay, what is epitope? Simple definition: special part, special part of antigen, special part of antigen attached with where? Attached with receptor, or attached with receptor, or attached with antibody. Okay, so that special part present on anti, I just give a example here. This part is much special. This part attach, or this part bind with this full length is antigen, but only this part have ability to bind. Okay, for example, this is the cell. Cell have receptor, cell having receptor. That antigen is the full length is antigen, but only one part can attach. Only here, the only this part can attach. Oh, that this special part known as epitope. Done. If you have one epitope, is okay. Or one epitope. If you have more than one epitope is known as valence, antigenic valence. Total number of antigenic determinants. What is antigenic determinants? Epitope is known as epitope. Same thing, okay? So number of epitope, number of is mean number of binding site, number of active site on antigen, number of active region. Understand? So what is epitope? It's the active region. It's a special part. It's a binding part. Yeah. Too many things about epitope. This is the property of antigen. Okay. So what is epitope? Okay, let's see what are the factor affecting immunogenicity. Why antigen? They have stimulating property. They, of course, they also have reactivity property. uh, Weak and weak and weak, small and small and small antigen have ability to reactivate. The main thing, stimulation of immune system. Okay, so antigen having property immunogenicity. Why they have property like immunogenicity? What are the property? Molecular size, size very important. Chemical structure complex. Okay. Antigen determinants. Antigen determinants. What are the antigen determinants? These are the epitopes. Okay. So factor from the recipient. If the totally outsider, totally different structure, of course, they can stimulate easily our immune system. Okay, have immunogenicity. If the structure more similar to our body, less property of immunogenicity. Okay, the genetic part of that antigen, the immunization method. Let me discuss this. Okay, let's see our chemical and physical property. Okay, let's see. If the molecule, the size of molecule 10,000 or 100,000, above 100,000 molecular weight, it means is smaller molecule, weak immunogen, more bigger molecule, stronger immunogen. If the antigen size is smaller than this, a bit weak antigen, not have immunogenicity much. Okay, not have immunogenicity much. If the size is bigger, you can see here. Okay, if the size is bigger, below this they are weak. If above this, they are more stronger immunogens. Okay, they stimulate. Stimulation is very important. Okay, can you do you remember your adaptive immunity for activation needs stimulation. Innate immunity, they can work without stimulation. Virus enter, bacteria enter, innate immunity start fighting. Innate immunity start fighting. But acquired immunity, very strong immunity, acquired. But the one problem with the acquired, what problem? Acquired immunity, they need stimulation. Acquired immunity, they need stimulation. Can antigen stimulate? Or if antigen can stimulate, then immune system can work more better. Okay, can work more better. So one property, molecular size. If antigen, okay, chemical structure, antigen protein or carbohydrate or fat or nucleic acid. If antigen is protein, I'll explain. Protein, Mostly antigen enter your body, their nature is protein. Okay, mostly antigen enter your body, stimulate your immune system. If the chemical structure is protein, they have property to stimulate. If the chemical structure is lipid, very weak property to stimulate. Understand? You can see here protein. Then next, carbohydrate. Carbohydrate have ability to stimulate but less ability as compared to protein. If the antigen nature is protein, if antigen nature is protein, then they have more ability to stimulate. Clear? So molecular another thing. Okay, protein, simple protein. If the amino acid, same amino acid there, homopolymer. Homo mean same, polymer. If the protein, arginine amino acid, arginine, in the protein, three amino acids same time. Okay, arginine, lysine, glycine, three different amino acids. So if we having hetero, if the structure is complex, if the structure not much complex, weak immunogen. If the structure is too much complex for antigen, strong immunogen. Understand? Protein, which one is more stronger antigen? Protein, carbohydrate, nucleic acid, lipid? Which one more stronger antigen? Can stimulate, can react?
Protein.
Protein. Okay, okay, let's see the protein. Okay, simple protein, the complex protein, more stronger antigen. Simple protein, the complex protein.
Complex.
Complex protein, if they have variety of amino acid, different types of amino acid there, too many types of amino acid there in the structure, so they are more stronger aminogens. Understand? They are more stronger aminogens. You know, microbes, they're very clever. Bacteria versus, they are very clever. What they're doing, they release some lipids. Oh, they release simple proteins. Oh, so then our immune system not fight well. Clear? They are very clever. They want to escape from our immune system. They want to escape from our immune system. Heteropolymer is property done. Third property, antigen determinant. Oh, how many epitopes there? Epitopes are small. I already explain what is epitope. Okay, they stimulate. If you have just one epitope, weak immunogen. If you have more epitope mean antigenic valence. It's a variety, okay? Antigenic valence. If you have too many one, this is one antigen, complete one antigen. One epitope here, another epitope here, another epitope here. These are the binding sites. These are special types, okay? The special structure of the antigen that interact with the antibody, that interact with the receptor of the cells. So if you have too many epitope, if you have too many epitope, you are a stronger aminogen. Okay, if the antigen having few are just one epitope, just few epitopes, it's mean you are a weak immunogens. Done? You are a weak immunogen. So number of epitopes, okay? Number of epitope. So number of epitopes according to, okay, next we see what's the recipient factors. The recipient factor, you are outsider, non-self, self antigen. Which one is more better? They can stimulate. Self and non-self. Non-self is come from outside. Okay. Non-self structure totally different. Non-self structure like this. Okay. Self structure like this. You can see it's a self protein. This structure protein, cell, this structure protein, non-self. If the protein structure, carbohydrate structure, totally different. Yeah, totally different. It's a foreignness. Foreignness. You are a totally outsider. I just give example. One country, they are living all people, white people. If one person from Africa going there, I think that person totally different. We can see, yeah, clear. If the one person from Japan enter to China, I think they are more similar. They're more sim. Can you recognize Japanese from Chinese in a given person? One Japanese, a bit difficult. Okay, if you place one African there in China, in Chinese, between Chinese persons, you easily can see, oh, that's African. It's a foreignness. Okay, it's the foreignness.
Yes.
It's the foreignness. So it's mean if the structure is very similar to body cells, they are weak. Okay, they are weak. Thank God they are weak. They are weak immunogens. They are weak immunogens. Okay, if the structure is totally different, thank God, the structure is totally different from our body cell. Then our body cell have more ability to recognize them. Then also have more ability to fight against them. Clear? So that's a foreignness. Foreignness, more foreignness, it's mean it's a totally outsider. Just give a simple example, black and white. Clear? Okay, one person from Africa, another person from Nigeria or Ghana, there who you play of same. Can you recognize who is Ghanaian, who is Nigerian? Of course, it's a bit difficult. Okay, if you place there in Africa one Chinese person, oh, it's a Chinese person, oh, it's totally foreigner. It's like that. Okay, so this is a foreignness. It's a foreignness. Okay, next thing, HL is a genetic. Genetic is also important when you're doing transplantation of organ. Okay, genetic. So why we need this genetic HLA genes? Okay, let I explain what are these HLA genes. HLA gene, there are two category HLA genes according to genes. Okay, there are one A, B, C, D genes and one DQR. There are two set of HLA genes. If the genetic makeup is same, more similar, then we can transplant easily. If this HLA totally different from each other, then we can't transplant organs. We can't transplant organ. Clear? So this we also is a it's a for force the same thing like foreignness. If the genetic makeup is a bit different, the structure of cells is totally different. If different, then rejection more early. If the structure very similar to the recipient, very similar, so that's why the who is the best donor, your twin brother or sister is your best donor for you. Twins brother or twin brother, because they grow from one zygote, because they grow from one zygote, same DNA, same structure, same DNA, same structure, HLA totally same. That's your best donor. Second, and any other best donor, yes, you try to find out in your family, because your family HLA very close to each other. Anyone else has same HLA? If you're going out of family, a bit hard for transplantation. Yeah, when you try to find out recipient for liver transplantation, donor for liver transplantation, kidney transplantation, the first we searching in our family first. In the family, who is preferable? If the twins, that's thank God, 100% accurate match for transplantation. Okay, if the twin, if they are twin. Okay, next close relative in your family, more better for transplantation. Why I can't take from other people if their HLA totally different. When you transplant kidney or liver, your immune system fight against that transplanted organ. Okay, and destroy that transplanted organ. So that's why in immunology, so this HLA information about this genetic of immune system is also important. Okay, if the HLA is totally different, then the cells are more immunogenic. Okay, more immunogenic. So if they are very close to each other, the structure HLA, the gene are more similar, very close to each other, then they are weak immunogens. So for transplantation, of course, we need what? Weak immunogens. Weak immunogen mean their HLA very similar, their body structure very similar, their cell structure very similar. Then the recipient immune system not fight against transplanted organ. Okay, so also it's one property. Immune system immunity less optimal, like it's a maternal IgG, age and health. In early age, in elder person, in newborn, the immunity is a bit weak. Okay, immunity, elder person less immunity, and in young person, young, just a newborn, they just building your immunity just start in newborn, and immunity just finishing in elder person, immunity at peak in young patient, immunity at peak in young patient. Okay, immunity at peak in young patient. So mostly hypersensitive diseases, mostly early in age, mostly early in age, not in elder person. Elder person already immunity is weaker. already immunity is weaker, more chance for infection. Immunity is weaker, okay. In early age also more chances for infection, because in early age immunity is just building up, the baby just building their own immunity, okay. It's take time, more chance for infection, okay. It's also age and health. The important property you guys keep in mind, very important property, size, okay, and their structure complexity and number of epitope, these three very important, okay. This third thing here, foreignness. So these from recipient, foreignness and genetic makeup, foreignness and genetic makeup, okay. This is all about antigen, okay. This is all about antigenic classification we also done, okay. This classification we also done, what's a complete antigen, what's a hapten. The remaining anything left in this chapter we will continue inshallah tomorrow, okay. We also see the next chapter tomorrow inshallah.
Okay, sir. Thank
It's
you.
enough for today class. Okay, assalamu alaikum.
Waalaikumsalam, sir.
Waalaikumsalam. Thank you, sir.